Good afternoon, everybody. I first want to preface my talk. After all that amazing science, I'm not showing any of my own science, which is unusual for me to give a talk without my own lab data or clinical data. And I feel a little awkward following all that great science. Also, if you're expecting a talk on FOG-001 and clinical data, none of that's going to be presented this afternoon. A lot of people say, oh, I can't wait to see the FOG-001 clinical data. It is embargoed to ESMO. So I'm really giving a talk on an overview of Wnt beta-catenin targeting in Desmoid, and just to present the study in itself. So I just want to lower expectations because everybody goes, oh, I can't wait to see the FOG talk. And there's very little FOG data. That's also an issue, and you'll see that we try to find whatever data existed with this compound. So with that background, I do have disclosures. Parabolis, I am on the ad board and a consultant for Parabolis for the clinical development. However, they paid none of my way here today, and they did not pay anything for this talk, just to be very clear about that. So Wnt beta-catenin signaling, I just thought I'd take this cartoon from this paper by Boo, published in Oncotarget a few weeks ago, to better understand the pathway so we can have a discussion around it. Now, in wild type, where there's no Wnt signaling, a complex exists between APC, axin, beta-catenin, and GSK, and this complex phosphorylates beta-catenin for degradation. Canonical Wnt activation involves Wnt activating, binding to the membrane, and axin moving up to the membrane, and releasing and breaking up this degradator complex. And now beta-catenin, it cannot be phosphorylated. Beta-catenin is released, it accumulates in the nucleus of the cell. It binds to TCF4 shown here and activates all sorts of Wnt pathways and the signaling pathway. Now, the pathways can be activated in cancer by two particular ways. The first is the FAP syndrome, which is now associated with colonic polyposis and desmoids. And in this syndrome, APC is mutated. And by losing this APC, there's loss of function of APC. It no longer forms the complex with axin, beta-catenin, and GSK3 beta. Beta-catenin now is released. Beta-catenin goes to the nucleus and then accumulates and transcriptionally activates the whole pathway. In desmoids also, there is the oncogenic event where beta-catenin is mutated. This is an activating mutation. In itself, it also is released from the complex. It no longer can be phosphorylated, no longer degraded. Degraded, moves to the nucleus, and then beta-catenin does its thing in the nucleus and activates this pathway. So in cancer and pathogenesis of the cancer in desmoids, we have the two processes, FAP, APC mutations. In desmoids, they're from FAP patients, a germline mutation, and then the spontaneous beta-catenin mutations that result in its intrinsic activation of this pathway. And NOS signaling, which is separate from this, seems to signal in some way to the same process at the very end. Now in desmoids, the spontaneous desmoids, APC of course is a germline defect, but beta-catenin mutations are found in exon 3, and there are two codons, exon 41 and 45. There are three patients shown here who have had desmoid tumors. One is at exon 45, S45F. There's also S45P in the lower panel, and there's also T41A in the codon 45, all with an exon 3. In the series published in Modern Pathology over a decade ago, they looked at whether this had any impact on relative differences between wild type and mutant. In this series of almost 250 odd patients, 92% of patients with desmoid tumors had a beta-catenin mutation, though there's some wild type patients as indicated here. Young group, which is pretty typical of the patient population we see. The mutation status was independent of site, size, and didn't matter how you obtained the tissue. One of the questions often come up is, well, does the mutation impact relapse-free survival? Is there a prognostic factor for desmoid relative to beta-catenin mutation expression? And there are multiple studies, a little bit controversial. On the study by Mullen on your left, he looked at beta-catenin mutations versus wild type, and he broke them down by codon exon site within the codon, and found that there was a trend in that the wild type patients did better, but then again, when he broke it down, there was no statistical significant difference between them, though the trend tended to show a mutation had a little less favorable outcome. In the second study, again, these are older studies by Desmoid, he looked at this between wild type and mutant. He grouped all the mutants together, and here there was a difference between outcome and progression, relapse-free survival, post-surgery between the wild type and the mutant. But again, it was barely significant, small sample size, smaller than the prior study. So it's a little bit up in the air where the mutation itself has some sort of prognostic significance relative to outcome after surgery in terms of relapse-free survival. And all these studies preceded the use of the current drugs we have available today. Now, the best preclinical data that I could find on targeting beta-catenin came out of the Ralph Pollock's lab several years ago, and he published in PLOS One with a drug called tegavivint, BC2059. This drug blocks the binding between beta-catenin and TBL1, which is transducin beta-like protein. TBL1 is important for binding of beta-catenin to the promoter, and it protects beta-catenin from degradation in this process by a ubiquitin ligase that can degrade beta-catenin. So in this study, he treated desmoid cell lines. Now, just tell you, desmoid cell lines are hard to grow, okay? They're really slow. They sit there on a plate, and you wonder if they're even, like, alive. So I've gotten all these cell lines from Pollock. I've tried to do some beta-catenin work in the lab by targeting some of the pathways. And you look at them and go, how can you even assay a drug? Because they just sit there. They just look at you, smile away, and, you know, it's hard to assay. But to his credit, he's established a really large bank, and we're doing some collaboration now on these, and it's a challenge. So in this panel, he was able to treat beta-catenin mutant desmoids, which is really, most studies are really an APC mutant colon, but this is a desmoid-specific study. And in culture, on the far left is a cell line that's a normal wild type, almost a fibroblast. But in the other three cell lines, all which have beta-catenin mutations, by using this drug that blocks this binding, he's able to show it in low nanomolar concentrations a dose dependency effect. On the bottom panel, he showed also that this drug, when put on a Boyden chamber, which is a chamber to look at tumor cell invasion through a matrix assay, these are nuclear pore filters, he's able to show that when compared to the control, which is on top, when he treated the cells in the chamber on the below, there was inhibition of invasion across almost all the cell lines, and he quantitated that on the below. You go to the upper right panel, he looked at the impact on TBL1 expression with this drug, and he found that when he IP'd it out and then looked at expression, he found that the drug at 100 nanomolars did generally decrease the expression of TBL1, explaining why this drug will decrease the beta-catenin process. And in the bottom panel, he then looked at beta-catenin itself and showed that in a Western blot, that by comparing cytoplasmic nuclear expression, then 100 nanomolars, it was dose dependent, there was loss of beta-catenin. So the mechanism is substantiated. It does indicate that by blocking beta-catenin in a desmoid tumor in a cell line, that you can impact growth and you can affect beta-catenin expression, at least by these series of experiments. Now, clinically, I think Wnt beta-catenin to me is the RAS of oncology. I mean, I think about the undruggable. How do you develop a drug against a very complex pathway? And I think it's sort of, once we do it, I think it's going to be not just affect desmoid, but could have impact across multiple tumor types. So this is a summary of where we are or where we have been with drug development over the years, starting with this drug, ventistumab. It's a monoclonal antibody. It targets the Frizzle receptor, which is upstream in this map here. It went through phase 1. There was some bone toxicity, mainly tumor fractures of the bone, and there was limited activity, and the drug has been abandoned. There's this drug FZD8FC is a fusion protein that blocks the Wnt ligand. Again, by this receptor signals into the nucleus to activate Wnt beta-catenin. Blocking this will block the pathway. Again, a common on-target effect appears to be bone-related side effects. And in this study, they actually gave zoledronic acid to diminish bone pain and fractures. It's the only study I could find in the literature that actually put desmoid patients on a phase 1. Most of these studies have colorectal cancer. I mean, everybody wants to cure colon cancer. I get it. It's a big market. Nice to see a desmoid patient on a clinical study. And they had 2 patients with stable disease at least 6 months. That was an encouraging sign. RxC-004, small molecule inhibitor of porcupine, protein serine alpha O palmitoyltransferase. Again, very high in the pathway. Again, on target, bone fragility. There's significant tox, GI, and fatigue, and there was lack of a single agent activity. And again, this other one, Wnt974, a porcupine inhibitor, again, high upstream in this pathway, GI tox, dysgeusia, and limited efficacy. Not very promising. So what about this new compound that we're talking about today? So FOG-001 targets the beta-catenin TCF4 interaction. TCF is a T cell factor that's important in binding a beta-catenin to its promoter. It also seems to protect it from degradation. And the idea is, can we block this particular pathway? Parabolis went through working with the Broad, worked on a cancer dependency map to find what was the cofactors that had the highest affinity for beta-catenin, and TCF4 had the highest affinity of all the cofactors that they could detect on a multiple high output screen. So they felt based on this binding, that would be a perfect opportunity to find a peptide that in fact would interrupt beta-catenin from TCF binding and then therefore lead to beta-catenin degradation and hopefully in the end prevent desmoid tumor growth. So this is just the codependency pathway that came out of the Broad. Looked at multiple cofactors. They did Pearson correlation relative to the binding to beta-catenin. And you see here the TCF-L or TCF factor had the highest codependency for all these factors. And this cartoon is a picture of the beta-catenin molecule showing the TC4 binding site. PCL9 is another binding factor, again, not as dominant. So it fits right in the center of this molecule and seemed to be an opportunity for therapeutic intervention. Like I say, it inhibits the beta-catenin directly at this TCF beta-catenin site, and it should allow for a drug development strategy by binding to the interruption between TCF, this factor, and beta-catenin. So it fits right there at this part of the beta-catenin molecule, displacing TCF4. Now, what makes this a little bit novel is the chemistry. These slides, by the way, were supplied by Parabolis, depending on their presentation. This is their piece just to explain how this works. It's a different chemistry. It's called a halicon, and there are several advantages of this molecule as opposed to more traditional drugs that block typical protein interactions. What are they? Well, here's the way it seems to work. It's a hyperstabilized peptide. It has a helical structure with amide bonds, which allows increased membrane penetration, increased membrane permeability, and hyperstabilization. So the chemistry is unique for this particular peptide as compared to other compounds that have been developed for beta-catenin blockade. Again, a little more about the compound. Unlike other peptides, which require a pocket, this seems to be binding to the surface of the molecule, so you don't need a classical pocket to fit into, giving some chemistry to its advantage over other small peptides, has high affinity, can be optimized, and it also has the ability to do E3 ligase degradation and proteasome degradation when linked to the target. So it's a next generation inhibitor and degrader, and these chemical elements seem to give us some favorable pharmacology over other drugs that have been developed against this particular pathway. So what do we know about this construct? Well, this is the experiments presented at 2 former meetings of ACR 2023 and 24 by C and his group from the company. I've taken these right off the poster. You can see here that when you compare a control, this Halicon 5, which is a control, negative control, versus the actual Halican-5, which is one of the TCF4 beta-catenin constructs, one of the first compounds that came out of their program. That if you look at the localization, that the new molecule, the Halican-5, as shown in the upper panel here, when the cells are treated, and this is COLO320DM. That's a colorectal tumor with both a P53 mutation and an APC mutation. It is not a desmoid tumor, okay? But it does have an APC mutation, so this cell line, this tumor would be one amenable to targeting the beta-catenin pathway. And you can see in the figure that by treating these cells in vitro, you prevent the localization of beta-catenin to the nucleus, whereas in the negative control, the beta-catenin, those green cells, they're all in the nucleus. And they then show also that the expression of beta-catenin goes down when this drug is added to the tumor cells. They then went on and looked at expression of genes that are regulated by beta-catenin after drug exposure, and one of them is called XIN2. XIN2, when treated, the expression goes down with treatment, and then there's the CXCL12. In fact, it's an activator of beta-catenin. Will suppress it when you block the pathway. The expression gets activated. Here, when you block the pathway, usually it's expressed and you block the pathway. Beta-catenin cannot do its thing and it gets decreased. So it functionally works as anticipated based on beta-catenin transcription, the quantitated. And then when it came to actually PDXs, again, there's not a desmoid tumor here, but colorectal, again, this model of the COLO320DM, the double mutant, you can see it in itself causes suppression, but remarkably in PDXs, they do get, that's the top panel here, and no weight loss. They do get inhibition of growth. And again, this is dose dependent, Q4 day. They saw an effect Q7 day dosing, and they had data also in ACCC that has a beta-catenin mutation, as shown here. I guess this is the closest we have to a desmoid with a mutation of beta-catenin, but no desmoid tumor. But this, I guess, would be the closest we have to a beta-catenin specific mutation with the treatment resulting in suppression of the tumor shown in these PDX models for ACC. So here it again, here is the FOG-001. It's this double mutant APCM TP53 showing that the drug does suppress quite explicitly the growth of a colorectal tumor that shares a P53 and an APC mutation. The clinical trial is now open nationally. We're actually running the study at Case, and we have opening at both the University Hospital and at Cleveland Clinic with Dale Shepard. The study is ongoing, and at this moment, this is the study design. It's open for 18 years and above, excellent ECOG status, exclusion criteria shown here, no bone metastases because of the concern for the effects on the bone, excluding patients with vertebral compression fractures, osteoporosis, inflammatory bowel disease, brain metastases. It's not just in desmoids, that is a big cohort, and we're most excited about that, but it's also open to any tumor type that the Wnt-expressing mutant tumor types that are identified in any next generation screen. Some tumor biopsies are planned, and future combinations are planned, especially in microsatellite stable colorectal and even in immunotherapy is a future consideration. But it is ongoing in desmoid. It'll be presented at ESMO, so I can't say anything about the study. I'd like to say something about the study, frankly, because I've seen data, but I can't, okay? So I have to leave it at that. Don't ask me any questions about study or Dale Shepard or anybody else who knows about the study, but it is I'm excited about it. I'm excited to present it, else I wouldn't be here today, frankly, and that's what we know. So that's where I stand, and I want to thank everybody for your time and open any questions you may have.
Gary, just out of curiosity, since TCF also seems to play a major role, have there been any attempts to target TCF in the past?
Right. I can't comment. I mean, they picked this up on the screen, and I guess there could be off-target effects if it's a commonly expressed factor, right? So how that impacts upon the toxicity, we'll have to see. It's a great question. I think anytime you go after one of these proteins that are ubiquitously expressed and affects transcription, you could end up having toxicity. And I have to say, just wait for the ESMO presentation. It'll answer all your questions, Chris. Trust me. And next year, if I get invited back, I will present the actual clinical data. I promise. Okay? I promise. Okay?
Maybe.
Yes.
Sorry. Oh, go ahead.
Okay. Just the helicons in general, so how do they behave in immunogenicity? Is there concerns there to deal with?
Not that I'm aware
Immune
of.
responses towards?
No, I don't. I mean, they're thinking about combining with immunotherapy, but I don't know for sure how immunologic it would be or what the immune functions would be. I mean, it's sort of T cell function, you're right? So it's a T cell rated factor. So it could, you're right, it could. I guess we should be concerned about that.
Yeah, but that's another TCF4. So the
T-cell
TCF4, there's two TCF4. This is non-immune related.
We'll have to wait for the see. I don't answer your question. I'm not sure.
Hello. Great talk. I'm excited to see the data on desmoid tumors. The mechanistic slide about how the disease develops is the story that we tell about beta-catenin being unable to be phosphorylated, and then therefore it accumulates and it gets activated. And that is also true of the S33 and S37 residues of beta-catenin, but we very rarely see those mutations in desmoid tumor patients. We almost always see T41 and S45. So I'm wondering if there's something in there that tells us about the mechanism of how desmoids develop and how we can treat it in a way that it would be more surgical than just blocking all of beta-catenin function.
Right.
So I don't know if that's something that Parabolis or the FOG team is looking into, trying to dissect all of these potential findings.
It makes sense. I think it's a great idea. And hopefully your data will share some light on that also. Right? But I think it's a great idea for more selectivity and maybe avoid some toxicity and give a more potent approach to the pathway.
Sounds good. Thank you.
Okay. Well, thank you very much. Thanks for inviting me.